Well, hello, this is an animation about the polymerase chain reaction. And what we're going to do, we're going to walk through this, and I'm going to point out some of the important characteristics of polymerase chain reaction. Basically, in the 80s, this process was invented by Dr. Kary Mullis, and he won the Nobel Prize for that. Basically, controlling DNA replication so you can copy a very specific region between two primers that you put in there. So this is much easier to explain when I just show you the animation. So we'll start out like this. So one thing to remember when you're thinking about DNA replication is that the polymerase reads the 3'5 direction, the 3'5 strand, and it writes a new 5'3. So that becomes clear when you take the mouse and run it over the strand of DNA, and one of these pops open. You can see that in carbon number 1, that's the base attached to there. Carbon number 2, carbon number 3, there's an OH there that the polymerase adds onto. 4 and 5, and there's the phosphate. So basically, if you look on this side of the molecule, that's that end right here that my mouse is pointing to. That's the 5' end. If you go all the way down to the other side here, this will be the 3' end. Therefore, down here is the 5' end, and down here is the 3' end. So as we go through this reaction, you will see that basically the polymerase does read the 3'5 and write the 5'3. So the next thing is, if you look, I've highlighted a couple of these strands. So you'll see it's a blue region and a red region. That's the region where primers are designed to be complementary to. And one thing to remember is that those sequences, the blue sequence and the red sequence, don't have anything to do with each other. They're not related in any way, shape, or form. They're just a distance away from each other. And what we're going to do by the polymerase chain reaction is copy the distance between those two primers, where those primers land. So let's start the reaction. The target will be, again, like I said, between those two primer sets. So at 94 degrees, this is what you use in a water bath or a thermal cycler. You heat it up to 94. It makes the DNA double strand become single stranded, or denature. Okay. We're going to cool it to 60 degrees, and the primers will land. Now you'll see it. The polymerase will read this strand and write this new one. So it's going to read the 3'5 direction and write this new 5 toward 3. Okay. So here comes the DNA polymerase. It's called Taq polymerase, or Thermus aquaticus bacteria is where it was found in. Okay. There it goes, reading the 3'5, writing the 5'3. Okay, that's the first cycle. So you went 94, 60, 72. So now we're going to repeat that. 94, it's going to unzip. 60 degrees, the primer is going to land. There's millions of copies of primers and millions of copies of everything here. And you're going to warm to 72 degrees, and the polymerase moves ahead. So now you're halfway there at the end of the second cycle. You see this one piece here, short to short, and this is short to short. So now we're going to heat it up to 94 degrees, cool it back down to 60 degrees, and warm it back up to 72 degrees, and the polymerase will complete its job, and now you have primer to primer. Now remember, in this polymerase chain reaction, you have in the tube that you're doing this in, you have lots and lots of copies of primers, lots and lots of copies of A's, T's, C's, and G's. You have a buffer in there to maintain the right pH. You have magnesium, which is a cofactor. You have DNA polymerase, and you have the template DNA that you're trying to copy from. You're making copies of. Okay, so let's go through another cycle. Fourth cycle, 94 degrees, 60 degrees, 72 degrees, and voila, now you're up to 8 copies. So you can imagine if you keep going and you have copies and copies and copies. So if you go one more cycle, bang, now you're up to 22. Well, the PB92 kit, we're going to have 40 cycles of PCR. So you can imagine this is only the 5th cycle and you're already up to 22 copies. If you keep doubling and doubling and doubling and doubling, you can see the potentials for millions and millions of copies of this target sequence. So that's how polymerase chain reaction works.